DNA is a spiraling chain-like molecule made up of 4 different...
We're going to pause and restart that. I just wanted to make sure that it was actually recording audio. And it is. Yay.
Stated Clearly presents... What is a chromosome? In previous animations, we learned that DNA is a spiraling chain-like molecule made up of 4 different types of nucleotides. Scientists have labeled them A, C, T, and G. We also learned that a gene is a long stretch of DNA, a specific sequence of A's, C's, T's, and G's that code for something. What does a gene code for? Usually a protein, or a group of proteins, though there are a few exceptions. In multicelled creatures like ourselves, proteins, which are coded for by genes, interact with other proteins and molecules to make up living cells.
Would you guys like subtitles on this? Yeah, okay. I forgot to put them on yesterday, I realized. Yeah, there we go.
Cells make up tissues, tissues make up organs, and organs ultimately make up entire living creatures. Individuals have different traits, in this case, differences in beak shape, largely because of differences or mutations in their genetic code.
Okay, so what we're going to do is we're going to take a look at these actual colors here. And we're going to say, okay, we've got these 2 finches. This is a finch, that's a finch. And they have certain things that are different. But most of it is the same because they are finches. And if we go down the side, it goes red, yellow, blue, green. Red, yellow, green. Red, yellow, blue, green, red, yellow. Oh, that's green. Why is that called a mutation? This is making me a little sad. Okay, never mind. Let me just see. Green, blue, red, green, green. Oh, yeah. Okay, this one looks better. I don't know what's up with that one. But we have here, we have green, blue, red, green, green. Green, blue, red, green, blue. So do you see how it's different at just one base? Everything's the same except for that one little base. That one little base is a mutation, okay? And how that happens, the DNA is going to get damaged in some way, that maybe the sugar phosphate backbone gets broken a little bit. And as the DNA goes in to repair that break, something gets substituted. We substitute an A for a U, or not an A for a U, an A for a C, or something like that. And that's a mutation, and that can lead to a totally new and different trait. If that trait is an advantage to those organisms, let's say that there were some nuts that had a really strong shell. Which of these birds do you think would do better breaking open strong-shelled nuts? The one with the bigger beak, because it's a stronger beak, it's going to have more crushing force. So if you have a mutation that leads to a bigger beak, then that bird is going to be able to survive better. It's going to be able to produce more offspring. And so that mutation will be carried on into the next generation and the next generation and so on, okay?
These differences can either cause changes in a specific protein's shape, and therefore its function, or mutations can change when or how much of a particular protein will be produced. Now that you clearly understand these 3 terms, and by the way, feel free to pause the video to study them if need be, it is now time to talk about chromosomes.
And don't worry, we will come back to these later, okay? So if you're not catching all this, we'll come back later.
A chromosome is an entire chain of DNA along with a group of stabilizing proteins.
Do you guys remember what some of those proteins are called? Pepsin. Pepsin is an enzyme. What do we call the stabilizing proteins in a chromosome, in the DNA itself? Starts with an H. Histones. Histones. When you get this X-shaped chromosome, there are actually even more stabilizing proteins, but we're not going to worry about those names, okay?
You may have seen images showing DNA trapped inside a chromosome, sort of like this. This is not how chromosomes hold their DNA.
That is wrong. It's not just a single strand along each side of the X. That's not what it's like.
If you were to unravel a chromosome, you would see that it's made of a thread.
What's that thread called? Chromatin.
Sort of like a spool of yarn. Zoom in again and you'll see that that fiber is a collection of protein orbs called histones, which are wrapped with a string-like structure.
What is that string-like structure wrapped around the histones? What is this thread that wraps around the histones? So this is, the chromatin is with the histones. Yes? No? The chromatin is with the histones. What is this string that actually does the wrapping around the histones? Shall we find out? Okay.
Zoom in on that string and you'll find that this is the chain of DNA.
So what is wrapping around the histones? The DNA wraps around the histones. And then once the histones are in there, the whole thing coils up and makes, what do we call this form? We call this form the chromatin. And then when it coils up even further, we get the chromosome. So we have the DNA. Let's go forward a little bit. The DNA wraps around the histones, which turns that DNA into chromatin. And then the chromatin coils up even further right before the cell divides into a chromosome. But if you were looking at a cell and it wasn't getting ready to divide, would you see these X-like structures? Or would you just see a bowl of spaghetti? You would just see a bowl of spaghetti. It would just be the chromatin. It would be like just loose yarn out all over the place.
This is the chain of DNA. It is extremely long, millions of nucleotides long, and often contains hundreds of genes along its length. In this image, we're looking at a chromosome in its condensed, yarn-like form, but...
So this is an analogy. When the chromosome is in its X-like shape, it is a lot like a ball of yarn where the yarn has been organized so that it's not just in a big mess. Does that make sense? That's what we've done, is we've coiled it all up so it's neatly organized. That's going to make it easier when the cell divides if it's neatly organized. Can you imagine if I took 46 balls of yarn and I unraveled them so they're just long strands, and then I took them all and I threw them onto the floor? How easy would it be to organize them to get one to one side and one to the other side? That would be very hard, yes? But what if instead I took 46 balls of yarn? Would that be easier to separate? Yes. And in fact, we'll learn later that it's not actually 46 balls of yarn. It is 46 times 2, 92 balls of yarn, but we'll get there.
Chromosomes are only packaged tightly like this when a cell is reproducing. This structure helps with durability as the cell is splitting in two. For most of a cell's life, however, chromosomes exist in a loose, noodle-like structure along with other chromosomes.
What form is that? What form of DNA is that? Long, thin, and noodly? Chromatin. And let's just watch what happened in that cell again. Let's watch cell division here.
Producing. This structure helps with durability as the cell is splitting in two. For most of a cell's life, however, chromosomes exist in a loose, noodle-like structure, along with other chromosomes safe inside the nucleus or centerpiece of a cell. Here is a photograph of an entire set of human chromosomes taken from a cell right as it was getting ready to reproduce.
So this is from a cell that is ready to divide. You'll notice that they don't really look like X's, and that's because stuff never looks like it really does in diagrams, okay? There are X's there, but they just don't really show up very well on the micrographs. Okay, so this is a picture taken under an electron microscope.
Your genome, the entire collection of genes that help make you who you are written across 23 pairs of chromosomes. That's 46 altogether. With very few exceptions, each cell in your body contains its own full copy of your chromosomes, your entire genetic code. Eyeball cells only use the eyeball cell genes. The rest are turned off. Liver cells only use liver cell genes. The rest are turned off, and so on for each cell type.
So every single cell in your body has all the DNA for all the functions. But do your eyeball cells turn on the liver genes and start making liver cells in your eyeball? Does that make any sense? Does it make sense for your eyeball to turn on liver genes? Okay, I'm going to assume you guys know the answer to that, or you're not paying attention, one or the other. Okay, because no, it makes absolutely no sense to have liver cell genes turned on in your eyeball. Okay, or can you imagine if we turned on the eyeball genes in your liver? You'd be down there looking at your intestines. Yes, that would be crazy. So you had a question or a comment? Yeah.
We asked a question.
Yeah.
But it has.
You do have liver genes in your eyeballs. They're just turned off. If they turn on, you would start having them behaving in ways that they're not supposed to behave. Yeah, I guess so.
Is there like stuff in the world that like turn on and
So sometimes what will happen, most commonly what will happen, is that if there is a mutation that tells certain genes to turn on when they're not supposed to be on, usually it's genes for things like cell division. Like cells are not supposed to just divide and divide and divide and divide and divide. Otherwise, you would just end up with a big mass of cells. And if that happens, can you guys guess what it is? If cells divide and divide and divide and divide and divide and divide and divide really quickly, and you end up with a big mass of cells that aren't really doing anything other than dividing. Does anyone know what that might be called? Sorry? That is a tumor, that is cancer. That's how cancer works, is that the sensors on the cell that say, stop dividing, we have enough cells right now, I'm touching another cell here, I'm touching another cell here, I don't need any more cells of this type in this location right now. Those sensors get turned off, and the division proteins get turned on, and it just starts making cells that don't actually go on to become liver cells. They just divide and divide and divide and divide, and then you would have cancer of the liver, for example. Yeah.
One member of each.
Sorry? A tumor is uncontrolled cell division. It's a whole bunch of extra cells you don't need. And what happens is they're so busy dividing that they never bother to go on and say, do liver things. They never go and make those liver proteins that are needed to function as a liver. They're just like undifferentiated cells. Yes? Why is it so deadly? Because what can happen is that, number one, it's going to start getting in the way of your regular body parts, right? So you can end up with a lot of pain. Number two, it can, if one of those cells breaks off and goes into your bloodstream, it can travel to other parts of the body, right? So if you have This is where we get into benign tumors and malignant tumors. Benign tumors is when the cells are not splitting off and the growth is not completely uncontrolled. You may get a benign tumor that you need to remove simply because it's getting in the way. Yeah? Sometimes people have a benign tumor in their brain and it's getting in the way of normal brain function because it's pressing up against parts of your brain and isn't allowing your brain to function as it should. But it's not shedding cancer cells, rapidly reproducing cells to other areas. So it's not spreading to other areas of the brain or other areas of the body. When cancer becomes malignant, that's when it starts, the little tumor starts shedding cells and they go into, say, the bloodstream and they travel to other areas, either other areas within the brain, if it were brain cancer, or even from the brain, it could spread to your lungs, to your liver, to your bones. Like there's all sorts of different places that cancer can spread. Usually it spreads within the same organ, but it will also spread to other places in the body if given enough time. Yeah. Not a very nice topic, but certainly one that's worth knowing about. One member of each pair of your chromosomes came from who? Mom. Where did the other one come from? Dad.
A pair of your chromosomes came from your mother, the other came from your father.
Not to like gender stereotype and make the mother's ones pink and the dad's ones blue, but apparently that's what we've done.
If you were to examine the two individuals of a pair, you'd find that for the most part, both contain the exact same genes in the exact same locations. If you were to zoom into the actual genetic code, however, you would find slight sequence variations between them. These are the results of mutations. Most of these mutations happened long ago and have been passed down from parent to child for many generations. Others, however, are completely unique to you. These mutations occurred either in the sperm or egg, which fused to make you, or they occurred early on in your development, making it into all or many of your cells as you matured. These unique mutations represent brand new genetic information.
So each person has 50 to 200 unique mutations. Those are ones that your parents didn't have. Those are ones that just happened either in the sperm or the egg, or that happened in the early development of the cells.
Sequences of DNA that may have never been seen before in the history of the cosmos. So, to sum things up, what exactly is a chromosome? A chromosome is an entire chain of DNA along with a group of stabilizing proteins. Humans typically have 23 pairs of chromosomes in each of their cells. That's 46 altogether. In your body, one member of each pair came from your father, the other from your mother. For most of a cell's life, chromosomes exist in a loose, noodle-like structure. Chromosomes are copied and then condensed like spools of yarn for durability when a cell prepares to reproduce by splitting in two. I'm John Perry, and that is a chromosome, stated clearly.
Okay. Let's go fill everything in. Oh my goodness. Are we writing this again? Ay. I feel like we've done this forever. What does DNA stand for? Deoxyribonucleic acid. I want to make that a capital N because the D and the N and the A should always be capitalized. What do we call these little monomers, these subunits? What's that? That is a nucleotide. We have A, we have C, we have G, and we have T. No. Sorry. Yeah, that's no. CG, and that one's going to be T. How do I know? Why am I getting all wound up about that? Why does T have to be the blue one if the red one's A? Because they're matching up. They're both pointy triangle ones. The C and the G are the round ones. In this example, What do we call this stretch of DNA? That is a gene. It is just a section of DNA. What does it code for? It codes for a protein. What does it code for? Usually a protein. You don't need to know this part, so I'm going to put it in brackets, or a group of proteins. This one you don't need to know. And you also don't need to know this one, but in case you're interested, that's called a ribozyme. I'm sorry, why did I put a Y in there? That should be an I. Anybody want to guess what a ribozyme is from its name? It's an enzyme made from, not made of ribosomes, but something really similar. Yes. Because if you look at the structure, it looks like RNA, yes? But again, you don't need to know this. But do you see how you can pick that out by looking for clues? You can say, I see the shape of this thing. I see its structure. I hear its name. It's got that "ime" at the end. It's got the ribo at the front. What would it logically make sense for it to be? So we have proteins. Proteins make up cells. Cells make up tissues. Tissues make up organs. Individuals have different traits. What is another word for traits? Characteristics. Traits are characteristics. In this case, differences in beak shape, largely because of differences in their genetic code. What do we call those differences? Mutations. Oh, and look, I fixed it, because you can see over here, do you see how this one's yellow and green and this one's blue and red? Those are different from each other. Mutations can either cause changes in a specific protein. What has changed here? Shape. If we change its shape, what have we changed, Samuel? If we change its shape, what have we changed? Function. Thank you, Jaden. Or, what is this little clock representing? We can change it's a clock when a particular protein is produced. That's okay. I know, it's so weird. It's so strange to me that lots of people don't use clocks. They just use their phone. But look, it's right there. What time is it? 110. Probably 111, but close enough. Or how much of a particular protein is produced. If you can't be quiet, please put your head down and take a nap, because we have a lot to cover in 5 minutes. Okay? A chromosome is an entire chain of DNA with a group of stabilizing proteins. If you unravel a chromosome, you would see that it is made of a thread. Thread. Much like a spool of yarn. So here we have our chromosome. Here we have the chromatin. Here we have the histones. And here we have the DNA. Zoom in again and you'll see that fiber is a collection of protein orbs called, what are those protein orbs called? Histones. Finally, zoom in on the string and you'll find that this is chain of DNA. It is extremely long, millions of nucleotides, long, and often contains hundreds of genes along its length. And we're going to do the rest of this another day. Your homework you're going to use the key or the video to fill in the rest of the blanks. How are you going to access the video? So that's going to be pages up to 116. So that's going to be pages 110 to 116. And before you start talking, where would you find the link to the video? The link is on page 10. Where did we start? 6? 106. There's a QR code. And remember, tomorrow I will not be here. I'm going to see my son's graduation ceremony at UBC. So you'll be having a work period tomorrow. All right, I'll leave some stuff in the book for you to do. Make sure you've got your cheat sheet so you can work on that as well. Okay, and then I will see you guys on Thursday. How well are you guys going to behave for the sub? Amazing. You'll have a TOC tomorrow. Yeah. It works. So ready to nap? I'm so ready for a nap, but I don't get to have one. It's sleepy time. Yes. Sadly, I have to get them ready for tomorrow too, so. Yeah. Oh, yeah, it's Flex.
It is what.
Right. Okay.
You get
I can. Well, if nobody wants any help.
You look, it's like I
I
cannot
know.
cheat from any way, man. Like
It's like the fortress.
yeah, it's weird. What if you cheat from the person in front of you? You brought yeah, I cannot. Like you see, there is also one too. You're gonna
Okay. What on earth? Oh, interesting.